Good afternoon, everyone. Welcome to our final session, presentation session of the MICD track. Today we have Xiaoqing Chang, who is going to be presenting to us on the application of PBK modeling in QIVIVE for prioritization of chemicals for toxicity testing. Thank you, Shaw-ching, for presenting here today. Without further ado, please take away the presentation.
Okay, so can you hear me okay?
We got you perfect.
Okay, hello everyone. My name is Xiaoqing Chang. I'm a computational toxicologist from Inotiv supporting NICETM contract. I'm very honored to be invited and glad to share my experience on applying PBK modeling and QIVIVE for prioritizing chemicals for toxicity testing. This is my disclaimer. Here's the outline of my talk. I will briefly touch on current trend of chemical safety evaluation, what's NICETM and its mission, how PBK models can facilitate in vitro to in vivo extrapolation and altogether put in vitro assay data into in vivo context. What are the key components of the QIVIVE process? Next, I will share some case studies and experience and lessons that we learned through conducting the studies. New approach methodologies, NAMs, including in vitro, in silico, and chemical-based methods that can be used to promote 3 Rs: reduce, refine, replace animal use in research and testing. Compared to animal studies, NAMs are quicker, more cost-effective, can provide mechanistic, more human-relevant information, and can also be easily adapted to high-throughput platform. However, before a NAM being accepted into regulatory decision-making, we need to determine how well an in vitro assay results can align with the in vivo outcomes. This is where the PBK modeling and the QIVIVE come into help. So in case you are not familiar with NICETM, I would like to introduce it. So NICETM stands for the National Toxicology Program Interagency Center for the Evaluation of Alternative Toxicology Methods. One of the key functions of NICETM is to develop and evaluate data from alternative test methods. It also provides operational and scientific support to QIVIVE. Through its website, NICETM provides information on relevant guidelines, accepted alternative methods, past and current QIVIVE and NICETM projects, meetings and workshops, and also providing computational support. In 2018, a strategical roadmap was published by QIVIVE to promote acceptance and adoption of the new validated methods. So the roadmap describes a framework for integrating 3 key processes that must be established moving forward. First is more efforts are needed to involve the end user, like regulators, industries, into the early development of the methods. Second is to develop more flexible and efficient approaches to establish confidence in new methods. Finally, to ensure that the method developer help educate the end users on the potential benefit offered by the new technologies so that the validated method can result in a product that regulators will accept and industry would like to adopt. So now let's go back to talk about QIVIVE. So this graph shows number of articles found in the literature with search term in vitro to in vivo extrapolation, or abbreviation QIVIVE, which clearly show a trend of increase in the number of published articles, especially in the past decade. During the literature search, we noted a mixed use of QIVIVE terms. Thus, I would like to make some clarification regarding the terminology before moving forward. So in broad definition, any approach utilizing in vitro data to predict in vivo phenomena or outcomes can be referred as QIVIVE. Thus, it's important to specify the context to which QIVIVE is applied to. In general, the term QIVIVE refers to 2 different aspects. Traditionally, the term QIVIVE is more commonly referring to extrapolation of those PK parameters from the in vitro experiment measurements after applying scaling factor. One of the most commonly used is to extrapolate hepatic clearance based on the in vitro intrinsic clearance method. And this is often an approach used for constructing a bottom-up PBK models. However, in recent decades, the term IVIV has been more commonly used to describe the process of converting an in vitro concentration associated with some bioactivity, such as AC50, AC10, to an equivalent external exposure level. In this case, IVIV refers to estimate in vivo dose corresponding to an in vitro activity concentration. The focus of this IVIV is on dose. We refer to this IVIV as IVIV of dosimetry. And in literature, some researcher also refers it as quantitative IVIV, QIVIVE, to distinguish from the IVIV of ADME parameter that we just talked about. We call it QIVIVE. And kinetic difference between the in vitro and in vivo systems shall be considered for this type of IVIV, for the dose. Therefore, pharmacokinetic analogies and implementation of PBK models are needed. One common application of PK models in pharmaceutical field is to predict the plasma or tissue concentration from a certain external dose, which is referred as forward dosimetry. While in the risk assessment field, often case is the people have the biologically monitoring data such as chemical levels in the blood or urine samples, and they want to know what's the plasma environmental exposure level leading to those internal levels. So this process is referred as reverse dosimetry. For QIVIVE, instead of using biomonitor data, we starting with in vitro activity concentration and then apply the reverse dosimetry to predict the corresponding external exposure levels that would leading to the internal concentration equal to that in vitro activity concentration. This graph shows how the reverse dosimetry works. So first, we set a fixed dose, such as 1 milligram per kilogram per day. And use the PK models, we can predict the plasma concentration at this dose. This plasma concentration can be Cmax and can be steady state concentration. And then by assuming a linear relationship between dose and the plasma concentration, we will be able to calculate the equivalent administered dose, EADs, corresponding to any target plasma concentration. However, we shall keep in mind that the linear assumption between the dose and the plasma concentration don't hold true for all cases, especially at a high dose, when enzyme saturation may likely happen. So this diagram summarizes the IVIV process that I just talked about. So we start from in vitro assay, from which we obtain an activity concentration using the dose response curve. From this activity concentration, we can derive the target plasma concentration. The most straightforward way to derive the target plasma concentration is to make it equal to the in vitro activity concentration. So this means you will assume whatever happened in vitro at that concentration, the same cellular response will happen in vivo. Then the reverse dosimetry can be performed to relate the target plasma concentration to an in vivo equivalent dose, from which you can derive the reference dose or margin of exposure and can be used for risk assessment. So the PBK models, they provide a relationship between the external dose and internal concentration. It's very important. So as you can see, this is a kind of complex process. So for each of the steps, depends on the study purpose and the data availability, you can have different choices. Like this which also bring variabilities. So the red stars added here to indicate those potential source of variabilities in the process. So like, for example, the in vitro assay selection, whether to use a non-specific assay, such as a cytotoxicity assay, or an assay measuring endpoints that's mechanistic relevant to the in vivo outcome that you are interested in. And when determining the in vitro activity concentration, which dose metric shall be used. Nominal concentration are often used. However, the nominal concentration do not necessarily represent the cellular or the free medium concentration. So we select a target plasma concentration, whether to use maximum or steady state concentration, and how to evaluate the uncertainty and variabilities in experimental measurements or the QSAR model predictions for those PK parameters. And also which type of PK model to choose. I mean, whether you use simple PK model or a PBK model with a complex structure, as well as the population variabilities. So these are the things that need to be considered when evaluating an IVIV outcome. So next, I'm going to show you how we performed IVIV to predict in vivo developmental toxicity for valproic acid and its analogs, while at the same time evaluating some variabilities, the impact of some variabilities that I just talked about. So according to CDC, approximately 3% of all babies born in the United States each year has birth defects. And many of these birth defects are caused by the pharmaceutical or environmental chemicals. So to screen large number of the chemicals, fast and cheaper methods are required. And we choose VPA because VPA is well known to be teratogenic in humans and animals. And at the same time, it's also used as a medicine to like anti-epileptic drug. The in vitro assay we select is a DivoTox Quick Predict assay, DivoTox assay. It's assay developed by Stamina is to screen the developmental toxicity potency. So it's a biomarker-based assay, and it measures changes of the ornithine and cysteine in the spent media after chemical exposure. So both metabolites are involved in metabolic pathways that are important for cell proliferation and differentiation during development. So the ratio of these two metabolites, OC ratio, is used as predictive of developmental toxicity potency. So this slide shows the typical dose response curve from the DivoTox assay. And one is for this OC ratio, the other is for cell viability. And the red line here indicates the level of change in OC ratio associated with a toxicity, developmental toxicity. So this is named as DivoTox threshold. So the cross points here between this response curve and the threshold lines are defined as a developmental toxicity potential concentration and also the toxicity potential concentration, which is related to the cell viability. And out of these 2 points, we choose the DTP concentration because it's a more sensitive endpoint. The early work has indicated that the teratogenic potency rankings using this DTP concentration correlated well with the results from in vivo studies. So the plus and negative signs show the positive and negative results in the in vivo rodent studies. Here we can see that VPA is the most potent based on the DTP concentration. VPA analogs with DTP concentration within 3-fold of that VPA were also developmental toxic in vivo. However, for those that had higher DTP concentrations, they had little or no in vivo effects. So basically, this showing this in vitro assay has very good correlation with the in vivo studies. So next step, we are going to build a PBPK models. So this table lists some examples of existing tools for PBPK modeling, including both commercial and open source tools. And depend on the availability and the usability, we selected guest class and an open source R package, HTTK, to build PBPK models for our studies. So HTTK is our package developed by EPA scientists. It provides a generic PBPK model structures and also require like minimum information to be able to build a chemical specific model. And this integrated chemical environmental ice is a web tool, like web resources developed by Nicetum. It houses curated data and also web-based tools. One of the tools is PBPK models, which actually containing the HTTK models, but it gave it a very user-friendly interface. So here are the two models we used. One is a simple one compartment pharmacokinetic model, and these equations that we use to calculate the steady state concentration. And then here is the structures of the HTTK PBTK model. And then we use Cmax when we use the HTTK model because Cmax is chosen for IVIVE as it provides the most conservative predictions. So this graph presents and compared EAD estimates using these two models. To evaluate the results, we compared EADs to the LEL's lowest effect level from rat studies and that we obtained from literature. So we can see for both models, they actually predict the LELs really well for one of the chemicals, but they are both over predicted for 3 chemicals and then one under predicted for APAs. But overall, of these two models, the HTTK PBTK model give more conservative and accurate estimate to the rat LELs, and for 4 out of the 5 analogs, EDs are using these HTTK models are within 4-fold of the rat LELs, so which we think is pretty good. Next, we apply the same approach to estimate the human exposure levels corresponding to the DTP concentration. And in addition to the previous PK models, we also use 2 pregnancy-specific models. One is an open source pregnancy model provided in HTTK-R package, and the other is the one built using the GastroPlus platform. For both models, we used maternal and fetal plasma Cmax as a target concentration. So this figure shows the EAD estimates using various human PK models and compared to the available human clinical data when it's available. So this circle represents the EAD estimate using HTTK models. So basically, we didn't see a big difference whether using regular, generic, or the pregnancy-specific models provided in this HTTK package. And we did notice that the EADs provide, predicted using GastroPlus model, provide a more conservative estimate across all the chemicals. So only for VPA, we were able to get the human clinical data. So basically, the human clinical dose that prescribed for the VPA, and which represents by this red asterisk and the plus signs, which provide a range. So you can see actually all the models and providing EADs that approximate to the minimal clinical dose of VPA. So this suggests that this clinical dose of VPA may exert some developmental toxicities when being used in pregnant women. So, and at the same time, because there are so many, abundant data for the VPA, we actually are able to get the PK data for the clinical PK data for the VPA, and then calculate the EADs based on that PK data represented by these dark triangles. And then we actually see there's very close estimate based on the gastro plus pregnancy models with maternal Cmax as target plasma concentration. And like I just mentioned, overall, the gastro plus pregnancy model with maternal Cmax as target plasma concentration give the most conservative EAD estimate for across all chemicals. In addition to evaluate impact of different PK models, we also want to know the impact of in vitro kinetics on the IVIV. As you know, even though it's a simple system, the in vitro assays, still there's a lot of different factors and process can influence the bioavailability of the test chemicals at the cellular target. So ARMTouch has developed a mass balance model to predict the chemical distribution based on the nominal concentration. So this figure just a theoretical representation of the concentration effect curve and corresponding to nominal, free, or the cellular concentration. So using the ARMTouch model, we were able to predict the free concentration from the nominal concentration and for all the VP analogs, and we also calculated the ratios between them. And compared to the nominal concentration, the free media concentrations are generally lower for all the VP analogs. As cell culture media, and then we use this free concentration to apply it to the IVIV and then compared to what we got from nominal concentration. So in general, and compared to the using nominal concentration, using the free concentration give a lower EAD across all chemicals, but the impact of this free concentration adjustment is chemical dependent. Like for example, this one, there's not much difference between using nominal and the free concentration, but here there is some difference. So in summary, the EAD estimates for the VP analogs based on different PK models were similar to in vivo data for both rats and humans. And the variations on the EADs between using different type of PK models are within expected range. And for the rat, we identified the HTTK models provided the most accurate overall predictions. For the rat, developmental toxicity LELs. For the human, the gastroplus pregnancy model with maternal Cmax as a target internal concentration provides the most conservative estimation, and also more accurate, the most accurate to the EADs predicted based on the clinic PK data. And the in vitro kinetic impact on these EAD estimates are chemical dependent, which is expected. So next, I would like to share some interesting observations on the nonlinear pharmacokinetic behaviors we observed in performing IVIV for a group of herbicides. As we have discussed earlier, we used PK models to establish a relationship between the external dose and internal concentration. Basically, it could be plasma Cmax, it could be tissue Cmax, or the AUCs. Depends on your interest. So the linear assumptions for this relationship has been commonly used. However, in some cases, in the relationship between external dose and the Cmax can be nonlinear. Here are 3 different patterns of the relationship between external dose and the plasma Cmax for the herbicide we studied. So for majority chemicals, it's still the linear assumption holds true, but for a few of them, we saw a nonlinear relationship when using gastroplasm models for modeling the high dose exposure and suggest some saturation occurring there with possible protein bindings or metabolism or some active transporting mechanism involved. So we then evaluate impact of this nonlinear relationship on the EAD estimates. So as the HTT models doesn't include Michaelis Menten kinetics or the transporter effects, so HTT model always generate a linear relationship between the external dose and the internal Cmax. So we noticed that the EAD estimated using HTT models are much smaller when using than those when using gastroplasm model for chemicals showing this nonlinear kinetics, which is not surprising because when you have a linear relationship and you would need less dose, to reach the designed target plasma concentration. So this suggests that even though the EAD prediction might not be accurate when using the linear assumption, but at least it can provide a conservative estimate, which is something desirable in risk assessment. Okay, so in conclusion, the QIVIVE is approach for putting in vitro toxicity data into in vivo setting. It is important in facilitated use and acceptance of NAMs in risk assessment. Our VP studies showed promising results with using in vitro assays that are mechanistic relevant to in vivo outcomes. Extrapolation with linear assumptions between external exposure and plasma concentration may not provide most accurate predictions, but can provide the conservative estimate for safe exposure in risk assessment. So challenges in the IVIVE is lying how to reduce uncertainty and better characterize variabilities to build confidence, which also creates a lot of future research opportunities. So finally, I would like to acknowledge many people who have been involved and helped on this project. This is the current NICETM group. Here is the website for the ICE, and we also have this is also a GitHub link for our open source QSAR model that you can be used to make predictions on chemical properties. Well, please feel free to visit them. Okay, thank you for attending the workshop, and be patient until the last session. And here is my email. Feel free to send me questions to my email if I didn't get a chance answering it live today.
Thank you, Xiaoqing, for a great presentation. We do have a little bit of time before we both head to the panel discussion here to answer a few questions. For your first case study example, they're asking, if you have any, in your observation, what are the potential reasons for the fold differences that you saw in the predictions of HTTK versus the GP pregnancy model?
Well, I think the main reason is the structure are different because the HTT version really is a very simple version of full compartment of PBK models, but the gastroplasm model, there's a lot of the things considered. For example, the the transporters, and then maybe the gut metabolism. So I didn't evaluate exactly what caused the difference, but yeah, I would assume those structure difference, and would make some of the contribution.
So potentially the model complexity or some of the other built-in pieces that were categorized in one model versus the other.
Yeah. And also, for example, like gastroplus, they are really, characterize the whole, the gut absorptions. But in the HTTP model, there's one factors, most of the cases, it just assume like 100% absorption. So
yeah, I was very happy to see some of the estimations that and observations from that comparison. Where it looked like the pregnancy model was working very well to help do those predictions.
Yeah, and for the pregnancy models, I mean, basically there's still a lot of structure difference. I mean, if you look at their structures and it's very different how they, the two models modeling the, the fetus and the placenta transfer are also different. Those are the factors that it's hard to be, to make it, apple to apple comparison between the two models. If those parameters actually wasn't available, I mean, there are no place to use in one of the models.
Very good. A couple more questions. I think we have a little bit of time here. The other one was, were you able to evaluate the sensitivity of input parameters affected by changing the dose metric in the second case study? In the case you have, would you share some of those observations?
We haven't done the sensitivity analysis. I mean, we did have some through our previous experience or studies. We did do some like local sensitivity analysis to study the impact of fraction unbound intrinsic clearance on the the outcomes. But for the second dose study, I haven't done the sensitivity analysis. Try to figure out what's the parameter actually to cause the non-linear kinetics. Yeah, I haven't done that.
Yeah,
but that's a good point. That's
one of those things that I think you highlighted in your conclusions too is the variability sometimes that we see and leads to some of that uncertainty as we do the modeling, and being able to better understand what those variability effects are as you use the different modeling types. Definitely a good question. And maybe just one more here, and then we'll switch back over. I want to be mindful of the time that we have. In the example, the relationship between Cmax and dose is sublinear. So linear assumption gives a conservative estimate. However, when Cmax and a dose is supralinear, linear assumption will be less conservative. That's, I guess, the question of that. Well, if the Cmax and dose is superlinear, will the linear assumption be less conservative?
Well, definitely it depends on curve. So just remember the presentation, the figure I show you is like the curve is like basically at the high dose. I mean, even you increase the dose, the Cmax doesn't increase. It just stuck there. I mean, so I would assume there's maybe some absorptions through gut absorption saturated. That could be possibilities. And of course, if the super then super linear, and then of course it will the opposite direction. You are right. It will make even less conservative for that case. Because because linear assumption is because we are really trying to make this method like high throughput, because we are really concerning screening chemicals, get quick results. So we want to do high throughput. That's why we really heavily rely on the linears. And for the non-linear kinetics, it just takes time. For examples, like for my case, I have to run it one by one using the GastroPlus. I haven't figured out a way to run batch. So basically, we just noticed like we actually using GastroPlus model, we detect some nonlinear kinetics, and this is what we detected. We haven't found chemicals that have the other way, so which is even conservative.
Yeah, that batching and that need to process many structures, is something that we always have an eye on here too. With between our ADMET predictor and our high throughput PK simulator, you kind of being that bridge until you get into GastroPlus and start moving through, and drilling down into more mechanistic models. So always happy to have different tools available for the different use cases, which is really the intent of the MICD track this year. Really is to show all of the differences in between traditional usage of modeling simulation in pharmaceutical development versus the chemical space. And I think here in the panel discussion next, we'll get into a lot more of how we as a community continue to take those next steps together to make sure that the modeling simulation is used across the board to do the risk assessments and all the rest of those pieces, but also for any unmet needs that are currently there with the tools that are available, and those how do we make sure that we work together to get that next generation of science and scientists who have that experience as just in a few of the talks today as well. Xiaoqing, I just want to thank you once again for a great presentation. Looking forward to the panel discussion here in a few minutes, and we'll see you there.
Yeah, thank you for the opportunity. Thank for everyone. Okay, see you in a little bit.